Good day, this is Professor Will, CBMD PhD. Today is Saturday, July 3rd, 2021. It is 11:45 and 52 seconds a.m. Eastern Standard Time. Today's research, Rett syndrome. So let's hop to it here. Uncovering the genetic mechanism behind Rett syndrome. Dysfunction in key gene causes neural stem cells to produce more astrocytes than neurons. Date July 2nd, 2021. Source, Kyushu University. Researchers found that the main gene that causes Rett syndrome, MECP2, controls the differentiation pattern of neural stem cells through the microRNA MIR199A. Dysfunction in MECP2 or MIR199A cause neural stem cells to produce more astrocytes than neurons. Furthermore, the researchers found that MIR199A mediates the production SMAD1, a downstream transcription factor of bone morphogenetic protein, BMP signaling. The researchers hope that the molecular pathway they identified, as reported in the Journal of Cell Reports, can lead to potential therapeutic targets for Rett syndrome in the future. Rett syndrome is a progressive neurodevelopmental disorder characterized by impairments in cognition and coordination, with varying severity, and occurs in roughly 1 in every 10,000 to 15,000 female births. However, it is difficult to initially identify because children appear to develop normally in the first 6 to 18 months. Rett syndrome is caused by mutations in a single gene called methyl CpG, binding protein 2, or MECP2. The gene was identified over 2 decades ago, and much has been uncovered since, but exactly how the mutations cause the pathology remains elusive. In their past research, the team had identified that MECP2 acts as a regulator for the processing of specific microRNAs to control the functions of neurons. And so, they went back to investigate if that pathway was also involved in the differentiation of neural stem cells. Compared to messenger RNA, the final template described from DNA that is used by a cell to synthesize proteins, microRNAs, or miRNAs, are much smaller and act to regulate messenger RNA to make sure the cell is making the correct amount of desired protein. And through research investigation, researchers found several microRNAs associated with MECP2, but only one affected the differentiation of neural stem cells, a microRNA called MIR199A. In fact, when either MECP2 or MIR199A are disrupted, researchers found that it increased the production of cells called astrocytes. Astrocytes are like the support cells of your brain, while neurons fire off the electrical signals. Astrocytes are there to help maintain everything else. During development, astrocytes and neurons are generated from the same type of stem cells, known as neural stem cells, where their production is carefully controlled. However, dysfunction in MECP2 or MIR199A causes these stem cells to produce more astrocytes than neurons. Further analysis showed that MIR199A targets the protein SMAD1, a transcription factor critical for proper cellular development. SMAD1 functions downstream of a pathway called BMP signaling, which is known to inhibit the production of neurons and facilitate the generation of astrocytes. And to investigate the process further, the team established a brain organoid culture, a 3D culture of neuron stem cells that can mimic aspects of brain development from iPS cells derived from patients with Rett syndrome. When they inhibited BMP, short for bone morphogenetic protein, the team was able to reduce abnormal neural stem cell differentiation. The research findings have given us valuable insight into the role of MECP2, MIR199A, and BMP signaling in the pathology of Rett syndrome. Further investigation is needed, but researchers hope this can lead to clinical treatments for Rett syndrome symptoms. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.